Having received feedback that the written instructions for several labs really weren't as great as they could have been, I'm recording a tutorial for the genetics lab. This will be initially useful for the Mendelian genetics lab, where we're going to talk about independent assortment and alleles and look at probability and look at corn cobs as examples of genetics. Next week, we're going to modify the syllabus, and I will update that on Canvas, that we will do human genetics. Trying to combine both sets of genetics problems into one lab was a bit much, so we will stick with Mendelian genetics initially, but the mechanisms that are going to go over in this lecture will be useful for how to set up genetic problems for human genetics. Vocabulary. This is the vocabulary list that is with your Mendelian genetics lab in the lab archives notebook. Diploid and haploid we already saw in meiosis. Just a reminder that diploid is a cell or organism that has its chromosomes in homologous pairs. For each type of chromosome, one from the maternal parent and one from the paternal parent. And haploid is going to be the gametes, having only one of each type of chromosome. Now, you're going to be asked about alleles and genotypes and phenotypes. A gene is the actual DNA sequence that's encoding for a specific trait. And in parentheses, I have an example. A gene for seed color. It doesn't say which seed color. A gene is just a characteristic. The allele is that specific variant of a gene. So if the gene is for seed color, the allele could be for purple seeds or yellow seeds or green seeds, whatever color is particular to the species we're looking at. So gene is generic, a characteristic. Allele is very specific. Exactly which variant of that gene are we looking at? The genotype is the type of alleles that are present. And we'll have examples of that. If you're asked for a genotype, you're being asked to provide which alleles are present in the organism. The phenotype is the physical appearance. So a genotype for here would be the letters that we use to represent the alleles for the seeds, and the phenotype would be based on the alleles that are present, what color do we actually see? We have both dominant and recessive alleles. A dominant allele is the one that will produce a particular characteristic that we can see even if other alleles are present. So that the recessive one is the one that has to be homozygous, having two identical alleles. So if you're a dominant allele, you can have one dominant allele and one recessive allele, and the physical appearance, the phenotype, will still be the dominant result. If you have two recessive alleles, then you're going to finally see the physical manifestation of that recessive trait. So two dominant alleles, you've only got dominant, you're going to see the dominant characteristic. Two recessive alleles, you've only got recessive alleles, you're going to see the recessive characteristic. But if you have one dominant and one recessive, the dominant allele dominates over the recessive one in terms of what it will look like. And it will look exactly like an organism that has two dominant alleles. When we're doing genetics problems, there could be lots of ways to do it, but the formalized, uniform way to do it so that your instructor understands that you understand what you're doing is to pick a letter to represent the characteristic, the trait. And we use uppercase letters to represent the dominant allele and lowercase letters to represent the recessive. So if we were studying seed shape and that smooth is the dominant characteristic, we would use an uppercase S for smooth and a lowercase s for wrinkled, not a W. Keep the same letter. We could pick any letter. I could have said X is for smooth, so uppercase X is the smooth allele and the lowercase x is the wrinkled allele. The reason for that is that once we get into doing genetics problems, we want to make sure we're looking at one trait, and so we have one letter that represents one trait. And that way, if we have two traits, we will only have two letters representing those two traits, and again, uppercase for the dominant characteristic and lowercase for the recessive. Because if we use two different letters, one for dominant and one for recessive, it's not too bad if we're only looking at one characteristic. But if we move on to two, then we're going to have four different letters, and it'll be extremely difficult to keep track of things. So pick a letter. Traditionally, the dominant allele is the first letter of the trait, but it could be anything as long as you define it. So uppercase S for smooth, lowercase s for wrinkled. Uppercase P for purple, lowercase p for yellow. So uppercase is dominant, lowercase is recessive for the same trait. And parents and the offspring are always diploid. We're going to be studying sexually reproducing species, and if they're sexually reproducing, they have to be diploid organisms so that their gametes are going to be haploid. So if they're diploid, every trait we look at has to have two alleles, two letters associated with it. Keeping with our smooth versus wrinkled, the parents or the offspring could be homozygous, two identical alleles, and dominant, so those identical alleles are both dominant. Heterozygous, one dominant and one recessive, because hetero means mixed, and this zygous suffix means in the zygote, when the two gametes fused and made that first cell, the zygote. So homozygous, the alleles are the same when we form that zygote. Heterozygous, the alleles are different when we form the zygote. And then we can also have homozygous, the alleles are identical when the gametes fuse to make a zygote, and recessive. We have to say if it's dominant or recessive if we describe the genotype as homozygous, because what are the two identical alleles? If it's heterozygous, we don't have to add anything because heterozygous means two different alleles, one dominant, one recessive. Parents and offspring always have to have those two alleles. Gametes, the reproductive cells, are going to be haploid, half the genetic information, so they have one allele per characteristic. So this SS, homozygous dominant parent, their gametes could be a big S or a big S. There's no other genetic variation. But this heterozygous parent, their gametes could be a dominant capital S allele or a recessive lowercase s. And then likewise, a homozygous recessive individual, they'd undergo meiosis, they would produce the cells, but they're genetically going to be all carrying that lowercase recessive allele. So the genotype is the alleles that are present and represented by the letters, and the phenotype is a description. So if the homozygous dominant, two big S's, that is the genotype, we would say that looks smooth. That's the phenotype. One big S, one little s is the heterozygous genotype. What does it look like? It's still smooth because that uppercase allele, the dominant allele, doesn't obliterate the recessive allele. It's still whole, intact, and genetically present, but its features don't get to be seen in the phenotype. And this mistyped it. It should be two lowercase s's. Let's just fix that with the pen. should be lowercase s, lowercase s, two lowercase s's. That is the homozygous recessive genotype. I want to get this back to the arrow. There we go. Giving us the wrinkled phenotype. When we're doing genetics problems, they are called crosses. And could have a monohybrid cross, which means we are crossing mating two individuals, and we're only following one trait. That's why it's mono. Hybrid is a mixture. We're mixing individuals, but we're only looking at one characteristic at a time. That makes it a lot more simple. Like if we try to discover or match all of the traits possible between even a simple mating, there's far too much to keep track of. We always start out looking at true breeding individuals, whether, well, with plants, I don't think you can get a true breeding human. That means they're homozygous. They have identical alleles for one trait. And an individual person could be true breeding for one trait, but they're not going to be homozygous for every single trait that's possible in the human genome. So if we're looking at one trait, the true breeding individuals are going to be homozygous. We'll look at a true breeding dominant, so homozygous dominant, two dominant alleles, and a homozygous recessive, meaning two recessive alleles. Those are the parents. The gametes that could be made, there's only one allele present. They could be a big S or that big S. This one can create a cell with a little s or another cell with a little s. Now we know that meiosis would undergo 2 rounds of divisions and there'd actually be 4 cells, but they're all going to be genetically identical, so we don't have to go through all 4. And then in a monohybrid cross, this grid that is set up is called a Punnett square because Dr. Punnett developed this mechanism for keeping track of alleles. We haven't determined which is the male and which is the female parent because for a characteristic that's not on a sex chromosome, it doesn't matter. So you could have said this is male and this one's female. A classmate might say this is female and that one's male. It doesn't matter. And we put the gametes along the edges, 1 gamete per box. And now we fertilize. So if this gamete fertilizes with this gamete, this one donates a dominant allele, this one dominates a recessive allele, and now we have an offspring that is back to being diploid, right? A haploid gamete and a haploid gamete fuse, they form a diploid zygote. Since these are both the same, these are both the same, the offspring, no matter which gamete fertilizes the other gamete, they're all going to have the same genotype. They're all going to get a dominant allele from this parent and a recessive allele from that parent. Anytime you mate a true breeding dominant, whether it's true breeding for 1 characteristic or 6, and a true breeding recessive for the same number of characteristics, the only possibility for the offspring is that they are going to be heterozygous. They get a dominant allele from the true breeding dominant parent, they get a recessive allele from the true breeding recessive parent, and they are heterozygous. So the genotype of this parent is homozygous dominant. Its phenotype is purple. The genotype of this parent is homozygous recessive. Its phenotype is green. And I kept the color coding in the offspring because we have 1 purple allele and 1 green allele in each seed. But because purple is dominant, this heterozygous seed that has 1 dominant and 1 recessive allele, its phenotype is going to be a purple seed, and it will look just as purple as its parent. We don't get any green seeds out of this mating. So the only genotype possible in a true breeding monohybrid cross, meaning a true breeding homozygous dominant individual mated with a true breeding homozygous recessive individual, will only produce heterozygous offspring. Because of that, the only phenotype possible is the dominant phenotype, which in the example we just used is purple seeds. And that is true for no matter how many traits. If we tracked 3 true breeding dominant traits crossed with an individual of 3 of the same true breeding but recessive traits, the offspring would be heterozygous for all 3 traits, and their phenotype would be dominant for all 3 traits. So there's the genotype that result. They are all smooth. So if we used, that was smooth and wrinkled, if we use purple and yellow, the genotype representing a corn seed that is heterozygous for seed color means you need to provide the letters that represent 1 dominant allele and 1 recessive allele. And in the phenotype, you're being asked to provide a description of the physical appearance. So the genotype of the corn seed that is heterozygous is a capital P and a lowercase p, because you're told it's heterozygous. You have to have a mix. The phenotype will be that it is purple, because even though there is a recessive allele present, the dominant purple allele masks that. The yellow allele is present. It is functional. It can be passed to offspring, but we don't see it in the phenotype. Now, if we cross in a monohybrid, mixing but following 1 characteristic, but this time, instead of true breeding, we have 2 heterozygous parents. So that's saying if we took the seeds from that first mating and then pollinated them, or just 2 unrelated seeds that happen to have this heterozygous genotype. So if we have a big S and a little s in the parent, the gametes could have either the dominant allele or the recessive allele, because 1 allele per characteristic in a gamete. And then the other parent has the same genotype, so 1 dominant allele in this gamete, 1 recessive allele in that gamete. And so if this gamete fertilizes with that gamete, the offspring get 2 dominant alleles. If this gamete fertilizes with that gamete, we get 1 dominant and 1 recessive allele in the offspring. The same here, a dominant and recessive gamete come together to create a heterozygous offspring. And if 2 recessive alleled gametes come together, the offspring is homozygous recessive. The genotypes, there are 3. We have 1 that is homozygous dominant, 2 are heterozygous, and 1 is homozygous recessive. The phenotypes, even though there's 3 genotypes, there's only 2 phenotypes, because both the homozygous dominant and the heterozygous are smooth. Only the homozygous recessive genotyped seed is wrinkled. So we could talk about ratios. The genotypic ratio is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. The phenotypic ratio is 3 smooth, because 1, 2, 3 genotypes give us smooth, and only 1 gives us wrinkled. A dihybrid cross means we're going to be looking at 2 characteristics at the same time. So we're going to look at color and shape. Purple is dominant, uppercase P. Yellow is recessive, lowercase p. Smooth is dominant, uppercase S. Wrinkled is recessive, lowercase s. So a true breeding dominant has to have 2 dominant alleles for color and 2 dominant alleles for shape. Likewise, the true breeding recessive has to have 2 recessive alleles for color and 2 recessive alleles for shape. There are 4 letters present, but there are only 2 traits, color and shape. We haven't made this person or this corn plant tetraploid. It is diploid for cell seed color. It is diploid for seed shape, just as we are diploid for every single characteristic in our cells. Figuring out the gametes when you've got 2 different traits is a little bit more complicated than when we only have 1 trait. So we have our homozygous dominant, purple is dominant and yellow is recessive, smooth is dominant and wrinkled is recessive. So if I number the letters, that will help you see how do I end up with the gametes that I do, because it's going to get more complicated when we get to the heterozygotes. So we have 2 different purple alleles, we have 2 different smooth alleles, we have 2 different yellow alleles, we have 2 different wrinkled alleles. So the first P could end up in a gamete with the first S, or the first P could end up with the second S. Let's see if I can manage to draw this with the pen and do a pen color. Let's do something that's a little different. So P1 could end up with S1, or P1 could end up with S2. We can't put P1 with P2 because then we have a gamete that has 0 alleles for seed shape, and we have to have a color and a shape allele in every gamete. Then how do we determine what the rest of them are? I'd really like to get my pen color different. Let's go with red. Then P2 could go with S1, or P2 could go with S2. So if P1 goes with S1, that's the gamete. If P1 goes with S2, there's the gamete. P2 with S1 or P2 with S2. We can't have 2 P alleles in a single gamete because that gamete would lack information for seed shape. We can't have 2 S alleles in a single gamete because that gamete would lack information for seed color. We would do the same thing for the recessive. We could put P1 with S1, P1 with S2, P2 with S1, P2 with S2. But when you look at those gametes, even though we have a different originating purple allele with a smooth allele, they're all purple and smooth. And even though we have a different yellow and wrinkled allele, they're all yellowed and wrinkled. So if we mate a true breeding purple smooth to a true breeding yellow wrinkled, no matter which one of these gametes fuses with which one of these gametes, we're going to get a big P and a little p. We're going to get a big S and a little s. All of the offspring, if we look at each trait separately, because there is a purple allele for color, the phenotype will be purple. Because there is a smooth allele for shape, the phenotype will be smooth. And so the phenotype for the offspring are purple and smooth. This is the same process as for the monohybrid cross. A true breeding dominant with a true breeding recessive, offspring have to be heterozygous. Just that now we have two characteristics. So they're heterozygous for shape and heterozygous for seed color. And now we are seed shape and seed color. And so we end up with purple and smooth. So that takes you through how to do the different types of crosses. The letters will change, but how you set up the problem for a monohybrid cross will always be a 2 by 2 Punnett square. A dihybrid cross, when we get to the heterozygous, is going to be a 4 by 4 cross. There will be examples in the lab archive notebook for you to carry those processes out.